Why do pathogens vary so much in virulence? And they do. Some cause infections with such mild symptoms that infected hosts are completely unaware that they are carrying the pathogen and transmitting it to others, whereas others may cause such severe disease symptoms that the host dies in short order. And indeed, the new coronavirus shows that variation within the human population. With some individuals, it now appears being infected and transmitting it without any awareness that they're infected, and others, as we've seen tragically, dying rapidly from severe viral pneumonias, respiratory failure, sometimes even kidney failure. There are basically three hypotheses that have been deployed in attempts to explain this variation. The first is often called coincidental evolution or accidental evolution. It's exemplified by tetanus, which can cause a really nasty disease in us, but we are not a natural host of the bacterium that lives in the soil, doesn't normally attack even any mammals. It's just a thing that happens. Short-sighted evolution is another possibility, and a plausible one in some cases. It's exemplified by the polio virus, apparently, which is, by the way, a close relative of the common cold. It lives in the intestine, occasionally invades the nervous system, adapts to growing there, causing the symptoms of polio, but it never transmits from there. So it was a mistake, as it were. It's an evolutionary dead end for the polio virus, but it happens, and it happens regularly. But if it weren't for that, polio wouldn't be a threat at all. And it's not part of polio's strategy in any larger sense. That's also the case for HIV, as we saw back at the beginning of this part of the course in Chapter 1 of Heron and Freeman, where they showed the evidence that within patient evolution of the HIV virus is what ends up indirectly killing people who come down with AIDS, but it's short-sighted, evidently, from the point of view of the virus. It doesn't actually contribute to transmission of HIV at all. Would be better if HIV didn't do it, but it happens. And finally, the hypothesis that's most interesting scientifically to us, because it involves real ecology and trade-offs, is the hypothesis that the costs and benefits of different virulence behaviors may affect the transmission rate of a pathogen under different circumstances. And a big finding from this line of thought has been that directly transmitted diseases, such as the common cold, need their hosts to be alive and mobile in order to be transmitted. And so all the many viruses that give us colds, which include rhinoviruses, the standard common cold viruses that are related to polio, but also some coronaviruses, not terribly closely related to the new one, but nonetheless other coronaviruses and various other families of viruses give us colds, but almost never cause serious disease, and the explanation is they've evolved to be mild because killing us is counter to their own long-term evolutionary success. On the other hand, vectored diseases, those carried around by insects or by water, may not have this problem and may do just fine, perhaps even better, by immobilizing the host and sometimes even killing it. So from the textbook, here are some nice data illustrating this last pattern. This is a comparison of different bacteria that cause diarrheal diseases. They're plotted on two axes, where the x-axis is the fraction of outbreaks that in which waterborne transmission is involved, and the y-axis is measure of virulence as deaths per infection. That is the percent of infected individuals who end up dying in certain data sets. And the pattern is a strong positive relationship between the two. These first 6 species never seem to normally involve more than 50% waterborne transmission, and they very seldom kill people. Whereas these 3, Salmonella typhi, the Shigella that causes dysentery, and the Vibrio that causes cholera, they are quite deadly, and in most outbreaks, waterborne transmission is a major factor. Here are data also from the textbook on other sets and types of pathogens that are vector-borne by insects or directly transmitted, and they show the same pattern. The histograms have sort of logarithmically scaled categories of deaths per infection. The right-hand red bars are 10 to 100% typical death rates, down to less than 1/10%. The green bars on the left are very rare mortality, and same pattern. The vector-borne pathogens are all over the place. Some mild, but quite a few of them causing significant amounts of mortality, whereas the directly transmitted control group, as it were, are overwhelmingly very non-virulent. That is, very seldom cause death. Well, it's a nice theory. It's been brilliantly promoted and developed by Paul Ewald, an evolutionary biologist who has convinced us that we like it. But what about an experimental test? So Jim Bull and 2 of his colleagues got the idea of testing directly the assumptions of the theory in a tabletop evolution experiment involving the bacterium E. coli and one of its viruses called phage F1. And so they just arranged a set of conditions that contrasted levels of opportunity for horizontal transmission, that is vectored transmission, between populations of unrelated bacteria. So in the limited vectoring treatment, they moved cells and phages between plates once every 8 days. In the frequent vectoring treatment, they did that every day, so 8 times as often. They provided the phage a chance to invade a population of previously uninfected and naive bacteria. And then they had their predictions based on Ewald's reasoning, which is that phages in the limited vectoring treatment should evolve to be relatively nice to their hosts, that is, less virulent, because their own fitness depends on the reproduction of their hosts to a greater degree than in the frequent horizontal transmission case, where the phage can, as it were, escape the devastation they've left behind in their previous plate. And did they get a result? Yes, a very strong one. The limited vectoring treatment led the phages to evolve lower levels of virulence, and they allowed the bacteria a higher growth rate. That's the X dimension, and a measure of virulence is the Y dimension. The two are sort of different ways of getting at the virulent phenotype. So they're a cloud down here in the lower left, whereas the frequent vectoring treatment led to much nastier, more aggressive behavior on the part of the phages. Okay, now let's leave that subject and turn to the way in which infectious diseases may be affecting genetic variation within our own human gene pool. The story here is that infectious diseases look as though they have been responsible, in the past anyway, for maintaining high frequencies of broken alleles, that is, enabling mutations of protein coding genes at quite a few different genetic loci. And you've seen an example of this already. Remember the story about how HIV uses this cell surface receptor protein called CCR5 as a co-receptor, part of its doorway, or I prefer to think of door handle, on its way into the cell. There are some people turned up early in the history of human HIV epidemic to be resistant naturally, and the cause turned out to be that they were homozygotes for a mutant form of this CCR5 called the delta 32 mutation. It's a delta for deletion, and here's a graph on a diagram of that part of the sequence of the gene and the protein, showing this the mutation deletes these 32 bases, which are not a multiple of 3, so it causes a frame shift mutation and a stop codon downstream, which terminates translation of the protein, so the resulting protein is only about 2/3 of full length and non-functional, and in a way that apparently doesn't allow HIV to use it as an entry point into the cell. The allele, the broken allele, the delta 32 mutation, is quite common, up to 15% in some European populations, and it's clearly been in those populations and at high frequencies for a long time, so HIV cannot possibly be the cause of that. What was? Was it drift, or was it selection by some other circumstance? And a related question is, when did the delta 32 allele first arise? Here's a map again from the textbook showing frequency contours for the most of the old world. It's extremely frequent around the Baltic, in Scandinavia, Germany, that part of Northern Europe, with frequencies that fall off continuously in all directions through the old world. Analysis of its linkage with alleles of other genes nearby on the same chromosome suggest that the mutation first occurred roughly 5,000 years ago, and the evidence and argument for that is in the textbook on these pages 310 and 11. There are 17 Bronze Age European skeletons recovered from Germany, and from which it was possible to get DNA and genotype them. And it turns out 4 of the 17 are heterozygous carriers of this delta 32 allele, giving an estimated allele frequency at that time, around 3,000 years ago, of 12%, which fits right into this distribution, as though it's been like this, at least in this part of Europe, for nearly 3,000 years, and got there within just a few thousand years of the origin of the mutation. So, on that evidence, it looks as though the delta 32 allele increased in frequency from nothing to of order 10% in maybe as little as 2,000 years. So we can easily use theory we already know to ask, is it plausible if this was just a neutral mutation, if this is an accident, that it happens to provide a certain kind of resistance and so on, and it just drifted to high frequency? Is that a plausible explanation? Could drift take a new neutral mutation from a low frequency, say less than 1 in 1,000 gene copies, up to, say, 10% in, say, 2,500 years, to be generous? Well, 2,500 years would be roughly 100 human generations. So the question is, will 100 generations get you there plausibly? The minimum population size that's thinkable for Northern Europe 5,000 years ago would presumably be something bigger than 10,000. So let's try a population of 10,000, which will have a relatively high rate of drift, higher than if the population is larger. So I just simulated that scenario, collecting 50 typical trajectories for a neutral mutation beginning at a frequency of 1 in 1,000 and still polymorphic after 100 generations in a population of size 10,000. And here are those trajectories. Notice none of them even reaches 2.5%, much less 10%, which would be 4 times as great up here near the top of the slide. So if the population size were around 10,000, could the delta 32 have drifted to an allele frequency of 0.1 in 2,500 years? Pretty much obviously the correct answer is no. That is not plausible. And what if the population size were substantially larger than 10,000, as seems most likely? Well, then it turns out the chance is even less that it would drift that high, because the rate of drift would be 10 times slower. So this young allele, assuming it is as young as the linkage data suggest, it simply hasn't had enough time to drift to as high a frequency as we find it in that part of Northern Europe, in even a modestly large population. So that suggests a history of positive selection. What caused the positive selection? It's conceivable that the delta 32 allele gave some protection against typhoid or some other infectious microbe, but the exact benefit is still not known, at least as far as I and the textbook are aware. The reason this case is worth reviewing is that it doesn't seem to be alone. There are a number of cases that look to be similar, of apparently young, broken genes with medically significant polymorphisms. CFTR, as we showed earlier in the course, doesn't it seem like years ago? But no, it was only a few weeks ago. It's a case. Hemoglobin S can be interpreted in that light. GBA Gaucher has alleles that look like that, and some of the several glucose 6-phosphate dehydrogenase deficiencies also have that look to them, and there are probably others. So these are mutant alleles that may have been positively selected, implying that they could be evolutionary responses to novel challenges that emerged in then modern life some time ago when we started living in cities, changing our diet, who knows what. In any event, this is an area that's quite fascinating, because it's medically important to understand why these bad genes are at such high frequencies in our population, and they also get at the nature of the trade-offs that drive adaptation of interactions between microbes and us. There are lots of other novel features of modern environments that also affect human health, and not through interaction with microbes, pathogenic or otherwise. For example, our diets are radically changed from those of the hunter-gatherers our ancestors were not all that long ago. Here's some data from the textbook showing how different modern diets are from typical hunter-gatherer diets that have, for example, in particular, much less fat and sugar in them. And we suffer from a great many diet-related diseases for which there is a lot of genetic variation. Why is there all of that genetic variation for those susceptibilities? Well, the answer in a catchphrase would be gene by environment interaction. That is to say, there may be it may be that genetic variation that was without consequence in the past, because the diets to which certain genotypes made you susceptible didn't yet exist, and then movement into a new man-made environment that has those characteristics then reveals this variation and makes it a heritable cause of variation in health. An analogy, also discussed in the textbook on pages 558 to 559 in the health chapter, concerns myopia, nearsightedness. It's highly heritable in modern societies. Identical twins are twice as concordant, that is, twice as likely to either be or not be nearsighted, as are dizygotic twins. And a wonderful story emerged a while back that Heron and Freeman retell about myopia among Inuit native peoples of the far north of North America, who now have high rates of myopia among the young, but their parents' generation didn't. Before 1935, it was basically unknown, and so wasn't heritable. If no one has it, right, there's no variation, and so there can't be heritable genetic variation for it. Now, it does exist and is variable, and there is heritable genetic variation for it. This is, if the standard interpretation is true, a lovely example of a change in the environment creating genetic variation for a condition. And the environment is, if you don't remember the story, is basically reading other extended forms of close focus work carried out by young children at a time when the eyes are still developing morphologically. And so the hypothesis is that the development of schools, of literacy, of electric lights, and now, of course, smartphones and other devices that very young people may spend hours staring at, focusing on closely, that's a novel environment for the development of eyes, something that didn't happen back when our ancestors were really living like hunter-gatherers. And that's the trigger then to which people, it turns out, are now differentially susceptible. So selection may be changing the frequencies of these more and less susceptible alleles at all such loci now by causing natural selection that in the past didn't happen because, again, the susceptibilities didn't matter. So this is, as I said, great examples of genotype by environment interaction and a reason why we all need to learn that concept and how to recognize situations in which it's applicable. But there's still more to health than infectious diseases and food. There's also the social environment, which may affect health profoundly and in lots of ways differ from social environments in our recent evolutionary history. And so it is increasingly suspected that, again, we've invented for ourselves an environment with new challenges in the social realm that create difficulties and variation, genetically affected phenotypic variation of various kinds. Kinship itself has public health implications in modern societies, as inclusive fitness theory might predict. One dramatic finding from a number of years ago by Margot Wilson and Martin Daly, a Canadian academic couple studying, in this case, in Canada, they found that infants and toddlers were 70 times more likely to be murdered by step-parents than by their own biological parents in otherwise equivalent Canadian families, that is, controlling for the obvious socioeconomic factors. And here's the graphs from the textbook that show these results for what the authors call natural parents, meaning biological genetic parents. These are rates of victimization per million child years of parent-child co-residence. The murders are very rare, only a few cases per million, and are spread out over ages from 0 to 8. The rates per child year of exposure by step parents are vastly higher, right? Over 600 cases per million child years, especially for infants and small children in the 3 to 5 year age range. An important caveat, please pay attention to this, is that the absolute risks are not high, as these figures might lead you to think. They're less than 1 in 1,000 per year, and most of the victims, by count, are killed by their actual natural biological parents, not by step parents. That's because most children live with their biological parents. But this pattern, to which Wilson and Daly were led by theory that suggested that genetic relatedness could be a risk factor for the youngest, most vulnerable children. Once they started looking quantitatively at it, they discovered this pattern that the relative risk per opportunity is vastly greater for stepchildren than for biological children. But again, to emphasize, it doesn't mean that it's absolutely dangerous to be living with a step parent. It's not. Right, so to put it in perspective, though, it is a big difference. Smokers are only 11 times more likely to die of lung cancer than non-smokers, and we accept that smoking is very risky. All right, what about other subtle effects of relatedness variation within families? Mark Flynn and his colleagues studied stress and illness among natural and stepchildren in a village in Dominica. They looked at levels of the stress hormone cortisol and discovered that it did predict how many days of school the children would miss owing to illness. And they noticed that the stepchildren had higher cortisol levels. That's shown in that bar graph. And genetic offspring tended to be ill on fewer days than step offspring, and remarkably ended up having more children of their own as young adults. So again, this is just identifying that being unrelated to the individual's parenting you, although it's not absolutely dangerous in the scheme of things, it turns out it is relatively much more dangerous than not being related to your parents. So a bit of biology to be aware of. Homicide in general is, of course, a very serious public health problem, much more often adult to adult than adult to child. And rates vary greatly, for example, among U.S. states, but also among neighborhoods, among countries in the world, among all sorts of other administrative subdivisions, but we'll focus here just on the data for states of the Union. And one pattern that people have been aware of for a long time is that there's a large average difference between southern and non-southern states. So one theory that's long been popular is, it's poverty, because there's more poverty historically in southern states than in non-southern states on average, but that doesn't seem to work. Here are the states plotted with the southern states as filled circles, the non-southern states as open circles. The y-axis is homicide rate. I think it's homicides per million or per 100,000 or something per year. And then a measure of average male incomes in those states on the x-axis. The income doesn't explain a thing. It's the poorest states don't have higher homicide rates than the richest states, but there is a big difference between southern and non-southern states. Another popular theory is there's a southern culture of honor that requires men to avenge insults to self or family, but that doesn't seem to have withstood testing either. So Daly and Wilson asked, well, what about men's perceptions of their economic prospects, which might map onto perceptions of fitness prospects, and the degree to which risk-taking and violence could help them get ahead. And they thought, well, income inequality might therefore affect how men saw the costs and benefits of taking big risks of the violent kind. And so could it be the case that the levels of income inequality in states or other neighborhoods that men live in might explain something about homicide rate? And it turns out that, in fact, the income inequality levels of the states, as measured by the so-called Gini coefficient, which is the standard measure of inequality that's used by economists in all contexts and has been for a long time, it explains fully half of the variation in homicide rates. The correlation here is 0.72, right? Very strong correlation between state levels of income inequality and average homicide rates in those states. And so now, right, it doesn't matter whether you're Southern or Northern. It matters what income inequality is. So here are two non-Southern states with relatively high levels of income inequality, and they have among the highest homicide rates. And right, there's some Southern states with relatively low levels of income inequality, and they have relatively low homicide rates. And indeed, Daly and Wilson pointed out repeatedly that no other social science variable anyone had thought to come up with and try had ever explained as much of the variation in homicide rates as income inequality easily explains. And it does so at all different scales, which I still think is an amazing pattern. So, for example, they did it for neighborhoods in Chicago, much smaller subpopulations than entire states. Works the same way. Neighborhoods with more income inequality have higher homicide rates, and that's much more explanatory than just the average income. So once again, at that small local scale, poverty versus affluence explains very little, but the distribution of affluence explains a lot. What about access to guns, you might say? That's always another popular theory. And what about violence on TV? Both of those also seem not to work very well. The Canadian provinces, which I've added to the pattern for states of the U.S., do on average have far lower homicide rates. Is it just because Canadians are so impossibly nice? Well, maybe, but why are they nice is the question. It turns out they have much less income inequality on average than American states do. Canadians own many fewer guns. That would fit with the pattern, but doesn't seem to be very explanatory. And they watch all the same TV. So if it was violence on TV, you'd think they would be just as violent as Americans, but they're not. So again, you have these popular theories that don't seem to explain the facts. That doesn't mean that the Wilson and Daly hypothesis is necessarily totally true or most of the truth or anything, but at the moment it's doing a better job than the alternatives. And what is it really saying? Is it saying that this is adaptive somehow because of income inequality? No, not at all. They are psychologists. They emphasize that their hypothesis would be that psychological adaptations to past human environments incline us to adjust perceptions and attitudes in certain ways that at that time, given the social structures and opportunities for applying violence or threat of violence to various social interactions, might have increased fitness then. They're not arguing that those impulses and perceptions are playing out in adaptive ways today in modern human societies at all. Right. Okay. But it certainly, this idea has led to some productive questioning. Whether it's a large part of the ultimate truth or not, we'll eventually find out. And of course, you might want to know where Utah is. It turns out we are an honorary province of Canada with respect to this pattern: low income inequality, low homicide rates, but high gun ownership rates. What about Arizona, a state with which we might compare ourselves in many ways, sort of a sister state? Much higher income inequality and higher homicide rates. Okay, so the summary of this rapid romp through a number of issues in evolution and human health. Certainly it does appear that there are many health-related issues that we can illuminate in some way, at least by evolutionary analysis. The properties of pathogens, their infectiousness, their virulence, and so on, have evolved, are still evolving very rapidly under the influence of knowable trade-offs, and identifying these processes may well help us to fight the pathogens, and it won't have escaped your attention that this is clearly going to be spectacularly true in the case of the novel coronavirus, where we can watch this evolution right under our noses as it's happening. Pathogens appear to exert strong positive selection on many mutations in our own genomes that offer, or have offered in the past, some degree of resistance, even if the mutations have other harmful side effects in homozygotes, but possibly also in heterozygotes. The human gene pool contains many alleles that cause differential susceptibility nowadays to what are often called the diseases of civilization and to other consequences of novel environments that we've created. This is a great example, apparently, of G by E interaction revealing, or as I like to say, creating genetic variation through a change in the environment, not a change in the gene pool. Right, so that's that point. The novel environments have created heritable genetic variation for susceptibilities to myopia and diabetes and all sorts of conditions that we could perhaps manage a bit more effectively if we understood a bit better how they came about. And it may be the case that in quite a few ways human psychology is adapted to past environments and may include tendencies that, again, could be better managed if they were better understood, assuming our goal is to maximize health for everyone, which, of course, it is. Okay, so that brings us to the end of this lecture and of the course. It's been an enormous pleasure for me. I hope everything goes well with you and you all stay healthy and happy. Bye.